As Renate introduced, my name is Jörn Horschig. I work here for a couple of years already, and I want to talk about data sharing, reporting, and statistical fallacies. That's a topic that we at Artinus and also I as a researcher think are very important. Yet we see very often that it goes wrong, mostly unwillingly and unknowingly. So I hope I can share a little bit of insights that helps everyone. So I would like to start with a paper that was introduced or that was published about 3 years ago called Brain-Computer Interface-Based Communication in the Completely Locked-In State. That paper used FNIRS and EEG and showed that completely locked-in ALS patients were able to communicate using a brain-computer interface with this technique. So how did they achieve that? First of all, sorry, this is a bit misplaced. This paper got quite a bit of attention. So it got more than 80,000 views. And I'm not sure if you know how many views any of your paper gets, but if it gets more than a couple of thousands, it's a lot, and 80,000 is actually really a lot. So what did they do? Well, they trained a classifier on FNIRS and also on EEG and also on EOG as a control to be sure that the completely locked-in patients really were completely locked in and were not able to move the eyes. They ask questions to the participants, and then using FNIRS, EEG or EOG, they tried to classify a yes/no answer from the questions. And using FNIRS, they were able to get a 70% classification rate, which is quite a lot given that these patients don't have any other means of communication. So that's quite remarkable. Consequently, there was a lot of media attention. So, for example, there was a headline in The Guardian saying groundbreaking system allows locked-in syndrome patients to communicate. Also on the blog of Ray Kurzweil, who's one of the probably most renowned scientists at Google, this made it to the headline. Reuters, a news agency, also had that. Even the NHS in the UK had a headline about that. Mashable, which is a bit more like a social media tech site, also had a headline. Singularity Hub, which is a bit more for the computer science and machine learning nerds website. Variable. So really everywhere. That's for Medium. So it made quite a lot of attention. Even The Sun, surprisingly, also had an article about that. They called it mind-bending. Locked-in syndrome sufferers use mind-reading tech to broadcast a shocking opinion on daughter's boyfriend. So what was so shocking about it? Well, the shocking thing was that at the request of the family, the researchers asked whether he, the patient, agrees that his daughter is marrying her boyfriend. Boyfriend was called Mario. And on 9 occasions, the answer was no. So the daughter was not allowed to marry a boyfriend, according to the father. So of course, that is quite shocking indeed. The article ends with reaching out to Mario. So in case you're Mario, please get in touch with Jasper and the Sun. So let's get back to the data on a more serious note. So here you can see a part of the data from patient F. On the left panel, so the left 4 graphs are the yes responses, the right 4 graphs are the no responses. And you can see quite a striking difference between the yes and no responses. The 4 different panels are 4 different regions. And these are, of course, FNIRS responses. So on the y-axis, you see the change in oxyhemoglobin. On the x-axis, you see time. And you see in these 4 occasions that it's pretty clear. So a yes elicits a positive response, a no elicits a negative response. So that's very nice. However, then about 1.5 or 2 years later, there was a response on that article titled "Questioning the Evidence for BCI-Based Communication with Completely Locked In." The article was written by a colleague of the authors, and he was actually questioning whether they were computing their statistics correctly. So on the left panel, you see the statistics as this author, Martin Spüler, claims should be correct. On the right, you see the same data, but computed differently. So what is the difference? The difference is simply in the order of how to average the data. So on the left panel, the first averaging was done over trials, then the channels were averaged, and then the channels were averaged. The left panel is what the original authors say they did. And there you can see nicely that for yes responses, you see a positive response, and you see a very small standard deviation across the response, whereas for no responses, you see a negative response, and the standard deviation was also very low. In the right panel, in contrast, you see what Martin Spieler says is the correct order of averaging, which is first across channels, then across trials, then across sessions. Because that way, he argues, you get the variance across sessions, not across channels. And the total response is the same. So still, yes responses go up and no responses go down in the oxyhemoglobin response. However, the variance across sessions is really much higher than the variance across channels. And this is, of course, a serious issue. So the question is, did the authors really make this mistake? Did they really do something wrong? And the other problem is, even if they did something wrong, in the end, they used a classifier. So maybe they did a mistake here, but the classifier was still correct. Was it? Well, so when I read this the first time, that was about 2009, I decided to go back to the original paper and read the methods. So the method in the original paper says this. I'm going to read it aloud for you. So to ascertain the difference between the average ISI of true and false sentences, t-tests were performed. T-test was performed separately for O2HB, EEG, and EUG signals acquired from all the sessions and across all channels in a session, averaged over many sessions varying slightly between patients. Furthermore, t-tests were also performed for each session between the ISI of all the 10 true sentences and all the 10 false sentences. Example, Berlin is the capital of France, Berlin is the capital of Germany, across different channels in a session. And the critical part is this. T-test was performed separately for O2HB, EEG, and EUG signals acquired from all the sessions and across all the channels in a session, averaged over many sessions varying slightly between patients. And to be quite honest, I have no idea what they actually did. I have no idea if they first averaged over the one or over the other dimension. And this is really not clear from the way they describe this. And that's one of the problems with the paper, that they don't describe clearly what they did. And therefore, of course, they open themselves up to these kind of critiques. So it's a bit unclear what they actually did. And I'm going to get back to this at the end of the talk. But this is just one example that happened to have evoke quite a media buzz, and therefore that's why I'm highlighting this. But there are many, many papers which have problems like these. There's a very nice paper from Sandra Nieuwenhuis, Birte Forstmann, and Erik-Jan Wagemakers in Nature Neuroscience, published, I think, 2000. I'm going to state that in a bit. At least I think 2011, and they did a literature review of several hundreds of papers, and they found about 78 papers in which the statistics were done correctly and 79 papers in which the statistics were done incorrectly, and they are highlighting these in this paper. And I'm going to now explain a little bit what the problems are, because they summarized very nicely what common problems in statistics are. And mostly it's just a twist in thoughts that we're thinking a bit differently about what statistics is actually telling us. Most often our intuition is a little bit off from what we actually did test. So one typical problem that, or one statistical mistake that many people do are comparable with this study. So in this study, there were two different groups of mice, the virally transduced mice and the control mice. And for each of these two mice, there were two different time periods of the experiment. There was a baseline condition, and then they had a period in which they had a stimulation, so that a photoinhibition condition. And in this graph, you see the P3 amplitude, so that's microvoltage, electrophysiological measurement. And you see that in black, you see the baseline measurements. In gray, you see the photoinhibition response. And you see that there apparently seems to be difference for the photoinhibition condition in the virally transduced mice. So what did the authors conclude? This is what the authors conclude. The effect of the optogenetic manipulation on P3 amplitude is larger in the virally transduced animals than the control animals. Which, if you look at this, might be true, but it's actually not what the data is showing. The data is showing that there is a difference in the virally transduced mice between the photoinhibition and the baseline condition, but it's not showing that there's a difference between the two groups of mice. And this is one kind of mistake that many people do. They do a certain statistical test, and then they deduce from the test something that they actually did not test. It could very well be in this case, but this is not what they did test. What they actually want to test is something like this. So they compute the difference between the baseline and the photoinhibition condition for the two different groups. They put that in a graph. This would be the graph. And then they need to make a statistical test of that. So there's an interaction effect that they need to test for. One other problem with this is also, if you see a graph like this, so you see averages of two groups, and you see some error bar around that. Are they different from each other, significantly different from each other? Well, very often our intuition here is mistaken. So first of all, it's very important to establish whether the error bars are actually standard deviation, standard error, confidence interval, variance, or whatever. It's very important to state that in the caption of the figure. And this makes a crucial difference. So the difference is that if you calculate so if you have this confidence interval for these two groups, then in fact now you see that the confidence intervals overlap, but this still can mean that there's a significant difference between the two groups. Whereas if this was the standard deviation or the standard error, it could be different. Especially standard error is usually very small because it's scaled by the number of observations you have. Standard deviation is usually very large. So it's very, very important to know what you're actually showing. And then it's also important that even if error bars overlap, it can still be a statistical difference between the two conditions. This is an example from a website called Statistics Done Wrong. I will get to that a bit more in this presentation, and it's a really nice website which also explains some of the statistical issues that people have, some counterintuitive conceptions, and explains that really nicely. Okay. So getting back to the paper from Wagemeesters and colleagues. Another mistake that many people do is related to this. So again, there are two different groups. I'm not an expert in what they actually did here. I'm just reading off what I read on the graph. So there was a fluoxetine group and a vehicle group. And they had a condition before the treatment. So they measured sniff time, it's in rats. And before the treatment, there was no significant difference between the two groups. And now you see after the treatment, they found a significant effect. However, what did they conclude? Well, they concluded that the acute fluoxetine treatment increased the social approach behavior as indexed by sniff time in our mouse model of depression. However, again, this is not what this graph is showing. This is, again, the wrong conclusion. What they do show is that there's a significant difference between the two groups. But what their statement is about is that there's an effect after the treatment compared to before the treatment. But this is not what they are showing here. And again, this is a slight difference in how you phrase it. And maybe it's also not what the authors exactly mean, what they wrote here, but still it is what they wrote. So they're still making the wrong conclusion. And I have to say, because I forgot to say that when I introduced this paper, so these are not papers from like unknown scientists, and this is just bashing on these scientists. These publications were Nature publications, Nature Neuroscience publications, right? So really, really high impact publications. And So they survived the review process, and even there, this was not caught. So this is not a trivial thing, and this is not easy. It sounds very easy if you can explain it well, but we are just all prone to these errors. A third issue that they found very often in papers is something like this. So here they had 3 different regions of the brain that they analyzed, and they measured the escape latency. So they measured in the anterhinal cortex and the perirhinal cortex and the postrhinal cortex. And what they conclude from this data is that the escape latency in the Morris Rotor maze was affected by lesions of the anterhinal cortex, but was spared by lesions of the perirhinal and postrhinal cortices, pointing to a specific role for the anterhinal cortex in spatial memory. So does this graph and their statistical analysis really support that? You might have guessed the answer. The answer is no. So what they actually did find, what this graph is showing, that there is a significant effect post-operatively for the anterhinal cortex. But that doesn't mean that that's a specific role for the anterhinal cortex in spatial memory. So they would have, again, needed to check for the interaction between the different regions in order to point to a specific role for this one spatial region. This is not what they did. So again, a very small difference in how they report it, but it has huge consequences for the conclusion. And the problem is not just, this is not just nitpicking, right? So this is just not, oh, you did something wrong, so I need to report this. If you make a wrong conclusion about your data and you base your whole future career based on this, or a new project proposal based on this, just because you made a wrong conclusion from your data, even if you did correct statistical tests, then you might spend years of your time tracing for something that is just not in your data originally, and you might, you might have wasted the time. So therefore, this is really important to realize. Of course, I'm also aware that statistics is not everything. So this is a very nice example where you see that the means and the standard deviation and the correlation, they're all the same. Yet if we just look at the data, if we just look at the pattern, we see that they are all different, and we can all explain them in our words much better than statistics can in statistical words. Still, statistics is very important. So let me take a big step back and talk a little bit about statistics. So the favorite that maybe everyone has is what does a p-value actually mean? So p-value is defined as the probability under the assumption of no effect and no difference for obtaining a result equal to or more extreme than that was actually observed. That is the definition. I'm sure that many of us have heard the definition over and over again and have heard that also in multiple ways. Still, it's often not very clear what it actually is. So I found a nice example again on this website called Statistics Done Wrong that explains very nicely and very intuitively what a p-value is, what a significance level is, and what statistical power is. So let me walk you through this example. So let's say each cell in this grid or each square in this grid is let's say a drug that we want to test. Because that's easy to understand, and that's very easy to just explain also to maybe to your family, right? So every square is a grid. Every square is a drug. The blue squares are drugs that work. This is, by the way, also, of course, very timely at the moment with the COVID situation, but let's just assume it's some drug. So there are 10 drugs that work, but you're unaware of this. You just have these 100 drugs that you want to test. Well, most clinical trials, as you might know, are not perfect. The statistical power says how many of these 10 drugs can you actually detect. A statistical power of 80% means that out of the 10 drugs that do work, you're able to detect 8 of them. And that's very good. So if you have statistical power of 80%, that means you can detect 8 of 10 working drugs. So this is a good test, is it? Okay, so we have something else called the significance level. And that's something that you as a researcher also have under control. So usually significance level alpha of 5% is taken. And 5% means that you accept that 5% of your tests are actually wrong. So they give the wrong results. That means out of the 100 tests that you do, you know that you detect 8 drugs that work correctly, but you also detect 5 drugs incorrectly. So you assume 5 of the drugs that you find are actually not working in reality, just because, well, chance, statistics. But what does this then mean? This means that if you run a test on these 100 different drugs, you will actually detect 13 drugs that work according to your test. And you know only 8 of them actually work and 5 of them don't work. So if you by chance take any of these drugs, then there's only an 8 divided by 13, so that's 62% chance that the drug actually works. And now suddenly it doesn't sound quite that remarkable anymore, right? So you usually think, oh, significance level of 5%, so I'm 95% sure that I'm correct. Well, not actually. So in this example, there will be only 62% correct. And this is still a good example. So there's another example, and this also fits quite nicely with Artenis, because you might know that besides NIRS devices, we also make phantoms for X-ray systems, especially for mammography. And coincidentally, on this website, there was an example about mammography. So suppose that about 0.8% of all women who get mammograms have breast cancer. So that means if you test 1,000 women, then only 8 of them do have breast cancer. The statistical power of a mammogram is about 90%. That means that of the 8 women that you test, there's a 90% chance that you detect breast cancer, meaning that 7 out of 8 you do detect if you do 1,000 tests. So that is, of course, quite nice, you might say. However, now there's the other side of the coin. There's the significance level. And from experience, we know that from those women who don't have breast cancer, about 7% will get a positive reading, which means out of the 1,000 people you test, out of the 1,000 women you test, 70 will get a positive reading, although they don't have breast cancer. And this is, of course, a big problem. So you now you made a test, you know that there are only 8 women who do have breast cancer, and you detect 7, so this is quite good. But also you detect 70 who don't have breast cancer, but the mammogram still indicates that they do have breast cancer. So actually only 7 out of 77 do have breast cancer. So the actual probability that your test is right is only 9%. And this is, of course, much lower than saying that your test is 90% correct, just because you detect 90% of the 8 people. And these are two sides, not exactly of the same coin, but they're quite important to realize that you do have both statistical power and the significance level, and you do have to keep that in mind, especially if you want to do more applied research. Then it's quite important because you will affect people's life with it. So I can really recommend reading through this website. It's based on a book that you can also buy. I didn't write the book, so it's not I'm not marketing me, but it's very nice to read and nicely intuitively written. Besides that, there's also a website or a course that I can recommend, and I have to say, personally, I always recommend this course, and I never did it myself. I started, but then I didn't have time to finishing it. It's called Improving Your Statistical Inference, from a professor, associate professor from the Technical University in Eindhoven in the Netherlands. And I came across the course because I read the blog of Daniel Larkens. That's the tutor of this course. And the blog is really nice. And he explains really, really nicely statistical notions and also problems. He also wrote a nice paper about Bayesian statistics, explaining, for example, using Harry Potter, what Bayesian statistics means. So he also can explain things very well. So it's not only that he knows it, he's also able to really share his knowledge. So that's why I recommend the course. Plus, it has a 4.9 of 5-star rating out of more than 500 ratings. So that's quite nice. So you might ask, what is actually the role of you as a researcher? Because of course you have your own domain, right? You have a certain skill set, and for most of you, just at the side, you also need to do statistics on your data, not only to be sure for yourself, but also to convince the reviewers and to convince readers that what you did and what you conclude from your data is actually correct. So do you have to do data massaging? That's how a colleague of mine, a former colleague of mine, framed it always. When she found that the data was not quite as what she saw in the data, she said, I just have to massage the data a little bit more. And while obviously this sounds a bit wrong, I think most of us know that we are all doing that, and it's also actually not wrong, because your data actually, there is something in your data, and you might always need to adjust the methods a little bit according to your data, according to your paradigm, according to new insights you get. You just have to see that there's of course, a gray area where you can change a little bit in your processing, and then you improve your data results. And of course, you can, hack your statistics also if you want to. And we all know that we live in this world of publish or perish. So we all know that we need to publish and we need to have good results. But of course, that can have dramatic consequences. So let's take this example, for example. So this is a summary that shows that everything we eat and everything we drink causes cancer, but also prevents cancer. So each of the red dots is one scientific study. So there's evidence for wine that it actually protects against cancer, but there's also evidence for wine that it actually causes cancer. So you could also say everybody dies because of cancer anyway. But what is the consequence of data massaging? So if you would really hack your way into it, that would basically mean that you bias the whole publication, the whole field into one direction. You would only want to, for example, publish what causes cancer. So now suddenly everything causes cancer, because everything that protects against cancer would be a null finding, something that's not published, something that people are not interested in, because, well, it's the bad result. It's not the result you're looking for. While for cancer, it's obvious that it's interesting that something can cause cancer and something can also prevent cancer. In many other research areas, it might not seem that obvious. So it might seem that suddenly you think, oh, this is a not significant result, so this is absolutely not interesting. So I'm just going to shove it away and put it aside and don't publish it at all. But that, of course, has consequences. So first of all, the biggest consequence is that people won't know about it, meaning that they might do the same study because they have the same idea, spend a lot of time, a lot of tax money again on that, and suddenly they come to the same conclusion as you did. Of course, it could also be that you had bad luck. Maybe there was some randomness involved and you do have a null result, but actually someone else doesn't believe it, tries to do the same study and finds a positive result. That would also be interesting, but maybe he wouldn't never had the idea if he wouldn't have read your study. So try to not be too pessimistic about your results that you actually do have. There are papers which focus on these negative results. So worst case, of course, is you publish in these papers, for example, Journal of Articles in support of the null hypothesis, or the Journal of Negative Results in Biomedicine. Another alternative for publishing negative results is because if you don't think it's that interesting and you don't want to spend more time on it and you're frustrated, you can also use a preprint publication service like bioRxiv, for example. You put it there, it won't get reviewed, you still have it out there, it can still get cited. You don't have to go through the review process if you don't feel like it, but at least it's out there. At least you there's no shame in showing that you did something and really spend a lot of time, and then the data just didn't show what you want, right? So that's not a big problem. And I would really encourage you to at least do a preprint publication, even if you don't have a significant result in your paper. So let's go back to this paper from the beginning. So what did actually go wrong here? So what happened here? I mean, there was this reply, the response on the paper, saying that they did the analysis wrong. And then in response to that, the authors again replied and said, well, we didn't do it wrongly. But what happened in the end? So these are the 4 big problems. So first of all, one problem was the data was shared incompletely. So obviously, this PLOS Biology is a good paper with a high impact factor, and the authors did publish or share their data. However, they didn't share all of the data. And the worst thing was that there were missing data and they couldn't retrieve the data. So that way, of course, it was not possible to reproduce the whole results that they have, all the analyses. Meaning that that's of course already a bit questionable. If you don't have the data, if you don't have your data management all right, then yeah, no one can really check if you did something wrong or if you did everything correct. And usually, I mean, usually no one is checking, right? But in this case, it did have a high impact. There was even a court rule in Germany obliging a health insurance to buy an NIRS device for another logged-in patient because of the findings of this group. So it does have a lot of consequences, especially in these high impact fields. Then, as I said, the analysis was also not properly described and couldn't be reproduced because of that. So I showed you the methods, and to me, it's absolutely not clear what they did. I'm sure that the authors didn't do anything on purpose wrongly, but they at least didn't describe fully correctly what they did. And then the last problem is there was some investigation that started and the authors were not fully supportive in the investigations. So as a consequence, the paper was retracted now. There was, I think, end of last year it was. And that was because both the university as also the Deutsche Forschungsgesellschaft, so the governmental research agency in Germany, both had an investigation in these authors in this paper, and they both came to the conclusion that there was indeed scientific misconduct done. And again, I don't want to blame these authors specifically for doing anything, right? This is just an example of what can happen, what can go wrong. If you don't have your data management if you don't do your data management correctly, and if you don't describe your analysis correctly. So it could be that it was intentional, but I actually doubt that it was intentional. So there was data missing, method description was incomplete. Still, it's scientific misconduct. So it was still not the way science should be done. And that's by far not the only paper that was retracted. So they are constantly their retraction of papers. So even if this happens to you, if you find an error in your analysis or in your manuscript, or after years of publication, you found out that actually something you did in the past was wrong, you can still retract your paper. So they also author-initiated retractions. I read that, I don't know, at least I read a couple of months back that also some Nobel laureates did retract papers. So it's a normal thing to do. It's no shame. Of course, it's not good if you don't support the investigations if they are running into you. So what I do think is important, what the authors could have done wrongly, what the authors could have done differently, is they could have simply shared their analysis script and of course share all the data. And I would also encourage actually everyone to do that, not only if you have a publication which has that much impact, but generally. The reason for this is that others may learn from you, but you may also learn from others. There might be people saying, oh, I see that you did this and that. Why did you do that? You could have done the analysis in a different way. And so with just a simple email, you might suddenly learn a new trick. Also, your data might be much more than what you think. So if you spend tax money, if you conduct a scientific study, then it could be that there's much more to investigate into your data. Maybe most, if not all of you know that there's so much more you could do with the data, but you just didn't have the time. So then why let it rot? Why let the data rot somewhere on a server or backup disk? If you share the data, the advantage is that it can be used by others, but also together with the analysis script, other people again can learn from you. And that also has a direct benefit for you, because people will get to know your name, people will get to know your paper, they will probably cite your paper, they will contact you. So you will have more interactions with people. So I think it's quite important. On Wikipedia, there's a list of neuroscience databases. The same or similar databases exist for other data. Also from the EU, for example, there's a platform for sharing data. And I think this becomes more and more important. Related to this, in the neuroimaging community, there's a new standard that is now currently, well, let's say, it's not obliged, but it's strongly encouraged for many, in many domains. That's called the BITS standard. It's an international initiative across modalities, meaning that it's not for NIRS, it's not for EEG only, it's for fMRI, for everything. There's now an initiative to extend it also to data from accelerometers, for example. The standard concerns itself with how to call data, what file formats to use for sharing, and how to incorporate metadata. And especially the latter is quite important, that very often there's data shared, but then people don't know what the experimental protocol is, what the participant data is, what is the age of the participants. And this is all specified in this BITS standard. They suggest how you should organize your data, what folder names you should give it to, where to place what data files. And there's currently an initiative going on for FNIRS also to include FNIRS in the BITS standard. At the moment, it does exist for electrophysiological measurement and for fMRI data. The file format that FNIRS BITS will use will probably be the shared FNIRS data format called Smurf. That is currently, it's not in development anymore, it has been released. There was an initiative led by the MIT under Professor David Bowers. It's an HDF5 file format, but that's just a technical detail that most of you don't need to know. At the moment, there are hardly any programs that are SNRF compliant, but I hope there will be in the future. So we will definitely implement support in our software for SNRF. I hope in future there will be much more, because this will be beneficial for the whole field, for the whole FNIRS community, because it allows sharing data more easily. For sharing data, of course, journals also try to encourage that more and more. So this is, for example, from a form that authors who submit to Nature get. You have to write a short summary, and then there's a checklist saying, well, I did this, I did that, it's not applicable. And that's also good for you as a researcher, just to check this list, even if you're not publishing in Nature, which most of us unfortunately are not. It's still important to go through the list and check whether you did all the things. So did I actually, I'll describe if my statistical tests are one-sided or two-sided. So it's just a small checklist that you check, and then you go back to your manuscript and say, oh, I actually didn't write that. I do a t-test, but I don't say if it's one or two-sided. So you just add these 3 words, and then suddenly, yeah, your manuscript is of much more value to those people who want to know your methods. It also says something about software and code. So what software did you use for data collection, for data analysis? Well, and finally, of course, there's also the intentional mislead case that can happen. So this here it says 2 years ago, but I think now it's 4 years ago, 5, 6, 7 years ago, probably 7 years ago. There's a Dutch scientist, I know it could be 3 years ago, because this is about when he published the book. There was a Dutch scientist called Diederik Stapel, who actually, well, he made up all of his data, most of his data. So he was a behavioral psychologist, and he said that he collected data by sitting on a bench and then observing people. But he fabricated the data in dozens of papers, and that was in the end found out. And in the Netherlands, that actually was really a big thing, and also in the media. So I know that people now actually use his last name, Stapel, as a verb. So you can say, I staple my data, or staple my data, meaning I make my data up. And he published a book about how he did that, why he did that. And of course, it's a bit problematic, right? So this guy intentionally misconducted science. So he did something intentionally wrong, and then now publishes a book and wants to make money with his story, of course. But there was there's a translation into English, and you can download that for free. And then you don't necessarily support him. But that's, of course, also something that can happen, that there are just people who fabricate data, unfortunately, but these are very few people, hopefully. So, but what can you actually do? Well, of course, you can always ask for help. So my actual advice is try to involve a statistician when it gets complicated. You cannot know everything. We at Athenas also don't know everything. So we are, if it gets complicated, we're also not the ones you can ask, but there are people who have studied that, who spend years, decades on statistics, who can really help you what to do and can make your statistics right. Also, you can read and learn independently of how old you are. For example, follow the course on statistical inference and just broaden your horizon, learn something. Because statistics is not easy. It sounds easy. And we as humans are inclined to use easy terms and simplify things because that's just much simpler and cognitively doesn't require that much effort. But apart from statistics, I would also like to encourage you to develop an intuition by looking at raw and processed data instead of only statistics. And on the other hand, don't trust your intuition too much and be critical. But I think the two have to go hand in hand. And there's a famous example that you maybe know. So if you plot the number of pirates in the world against the global average temperature, you see that the less pirates there are, the higher global average temperature actually got. So you could probably reduce from that we just need more pirates in order to fight global warming. But if you think about it, it's cost total nonsense. So that's not something you should actually do. There's a nice webpage called Spurious Correlations, where researchers gather all these things that correlate with each other, which have absolutely nothing to do. So that's also very, very nice, just to browse through and to have a laugh about silly things that people do. But then also keep in mind that you might also do something like that with your own data, where you actually don't realize that it doesn't really make sense. And just because you find a correlation between two things doesn't mean that they are related with each other. So that's pretty important to keep in mind. And finally, show raw and processed data in your figures. So I think it's very important to show also the raw data so that people know how did the data originally look like. And then you show the data how you process the data so that they know what you made of it. Share your knowledge with fellow scientists and share your data and protocols. That's also pretty important. And finally, of course, just in case, if you're Mario, then please get in touch with Jasper. I'm pretty sure they're still looking for you. Thanks a lot.